Here's a visual illustration of sequence space at various levels. These are projection of hyperdimensional sequence space onto 3 dimensions. So imagine projecting 3 dimensions onto 2 dimensions. Do the 2-dimensional projection of shadows of 3-dimensional objects look closer together? They look closer together because let's say I had a bunch of balloons in this room and then I had a light behind them, and I projected it onto a board, the shadows of them. Would the shadows look closer together? Yes. This is projection of thousands or hundreds of dimensions onto 3 dimensions. So these balls look closer together than they really are in sequence space. But you notice something from going for less than 30 amino acids to proteins with greater than 300 amino acids. Are these closer together in space and compared to these ones? On average, the distance increases in a linear manner as the size of the proteins increase known protein systems. So as the size of the linear distance increases, what happens to the random walk distance on average? It increases exponentially, right? So that's the statistical argument against evolution from my perspective. For me, that's the most convincing argument that nails the coffin. The nail's the last nail in the coffin for evolution. For me, that's what convinced me. But there's other arguments like God wouldn't have done it that way. This guy, Francis Aya, he's a professor of biological sciences, National Medical Science Laureate. This is his argument against the God wouldn't have done it that way argument.
The implication of intelligent design is that God is a very, very bad engineer. I mean, an engineer who have designed an eye with the optic nerve having to cross the retina would be fired. An engineer that would have designed the human jaw would be fired. Our jaw is not big enough for all our teeth. God making this trivial, obvious mistake and errors of design, well, maybe their God does those things, certainly not mine. I don't want to have to worship a God that is not smart enough to do as well as a human engineer.
Okay, so, I mean, it's reasonable. Why would we want to serve a God who can't do it as good as you can do it? Okay, so the question is, how good can you do it? Okay, so here's the design argument for the human eye. You know, God just wouldn't have done it that way because the human eye happens to be wired backward. Okay, so here's the argument for the backward wiring of the human eye. Any engineer, according to Dawkins again, would naturally assume that the photocells would be pointed toward the source of light with their wires leading backward toward the brain. He would laugh at any suggestion that the photocells might point away from the light with their wires departing on the side nearest the light. Yet this is exactly what happens in all vertebrate retinas. Each photocell is, in effect, wired backwards, with its wire sticking out the side nearest the light. The wire has to travel over the surface of the retina to a point where it dives through a hole in the retina, so-called the blind spot, to join the optic nerve. This means that the light, instead of being granted an unrestricted passage to the photocells, has to pass through a forest of connecting wires, presumably suffering at least some attenuation and distortion. Actually, probably not much, but still, it's the principle of the thing that would offend any tiny-minded engineer. So let's see this. This is the verted retina versus the inverted retina. And this is what, like arthropods and squid and octopus, they have this wire the proper way, where the photocells point toward the source of light and the wires stick out the backside. Our retina and mammals' retina are wired with the wires in front of the source of light, so the light has to go through the wires, then all the way to the back, and here's the detection apparatus here, and this is the back of the retina, and so this is where the detection of light in our retinas is, and so it's wired backward. Who on earth would design such a backward, fumbled up concept, right? Certainly not an intelligent God. So, but there's certain advantages to having an inverted retina. It allows for greater visual acuity and image processing power versus a verted form. The cost is high for high visual acuity. For example, the human retina uses more energy than the brain's cortex, 300% more. It uses 50% more than the renal cortex and 600% more than the heart. Okay? So in order to provide that kind of energy for visual processing power, you have to provide a great deal of nutrition. And so you need to put the photocells, or the source of high energy utilization, close to the source of energy supply. Okay, so they need to be closer for that part alone. Well, there's another problem with it. Also, for the squid, for example, if you have your rods and cones pointed toward the source of light, which direction do the little discs on the rods and cones slough off of? The tip, right? Sloughs off the tip. If they sloughed off the tip at a very high ratio, because we slough off thousands and thousands per day, it would cloud up your vision in front of the cone if it's sloughed off in front of your visual field. If it's turned around and it sloughs off into the choroid plexus, it can be absorbed and not mess up your visual field. Okay? So those are two things. What about the whole distortion idea? The wires happen to be translucent, but even then, translucent wires can distort light a little bit. It was recently found in 2007 that there are these cells that penetrate all the layers and they act like optic fibers, like fiber optics. And they take the light from one side and they go through by fiber optic transmission to the other side and then release the light undistorted. And so this solves the whole distortion problem. And who would have guessed it? Except by intelligent design. This is the inner life of the cell. These are white blood cells moving along the vessels. And then they come to a source of infection. This is the white cell rolling along. And when they find the source of infection, all these little signals are put out. And then they attach there, and then they go through the vessel wall. These are little islands that support proteins on the outer surface of the cell, and they do signal transduction. They float along with the protein sticking out. This is this external skeleton of the cell, made up of proteins and these islands floating around. This is traveling in through the skeleton of the cell into the inner cytoplasm of the cell. This is all put out by Harvard, by the way. You can look up the video online. These are microfibers that assemble themselves to a certain distance, and then they're modified in length by other proteins that come along at just the right spot and trim them to the proper length. This is a microtubule that comes along, and it's formed also like an amoeba needs to move, and it needs to form these microtubules. And then at the other end, where it needs to pull up its backside, it degrades the microtubules. And then you got these other things that walk along the microtubules and pull these vesicles for transport to different locations where you need to put other stuff. And it walks along just like that with two little feet. These are nuclear pores on the nucleus of the cell, and they spit out the microRNA like this when it's formed. And then the decoding apparatus attached to it, and they spit out the protein very rapidly. Then the proteins go from modification to the Golgi apparatus. And then the Golgi, see that it spits into the rough apparatus reticulum, and then it's modified in the Golgi apparatus. And then the proteins get put into these little vesicles, and then the vesicles get transported by the little walking machine to either outside the cell or other places inside the cell where they need to go. See how they're walking along, all these little blobs, when they're modified by the Golgi apparatus. And then they're spit out the cell, all the extra debris that it doesn't need. This is how the macrophage knows where to go. And it slips through the cell, through the wall of the vessel to go and attack the foreign invaders at just the right spot. Amazing, huh? And I think this is DNA replication. This is the DNA comes in double-stranded, and it splits off into two strands, and each one is copied. And see how the little arms of the apparatus copy each strand, and this one reaches just at the right spot to copy it, produces these oxidizing fragments. And this little apparatus by itself is extraordinarily complicated. This is DNA transcription. That was DNA replication, before. This is transcription into messenger RNA. It takes a bunch of proteins to connect to it at just the right spot. Then this thing gets released, and it flies along very, very rapidly, making messenger RNA. Incredible, huh? Just zipping along, and that's about how fast it goes. And then this is the flagellum in the end. I already showed you that. So that's all I have. Thank you very much.